All right, I'm going to get started, and we're actually only going to do basal ganglia today. The vascular component that I had planned to cover really relies on having covered the prior vasculature that we didn't get to on Wednesday. I'll try and put all of that together in a sort of video to watch offline. I'll try and get it done over the weekend, assuming both I feel better and also that we have electricity at home, which we haven't had for a day and a half now. So what we're going to do now is think about a different modulatory circuit. Okay? So the basal ganglia sit, and we typically think of them in the context of motor, because often motor is an easy way to study things. But as we'll try and show you over the course of the morning, it is far more than just the simplest motor that you consider, the how do I move my arm, how do I move my feet, and so on. But it's going to sit as a loop that interacts with cortex, where we know we have motor planning, we know we have motor output, but we also have decisions about when and whether to move. We know that thalamus gates cortex in a lot of ways. It determines what cortex is aware of and sees. And so the loop that we're going to build is one that puts a whole series of additional structures intermediate between cortex and thalamus. And there is already a reciprocal connection, the big one from thalamus to cortex, and a relatively moderate one from cortex to thalamus. But there's this additional loop that's going to be critically important. And we'll see that there will be some components, such as cerebellar function, which are going to come together and feed into both the thalamus, or cerebellum we know feeds into thalamus, but basal ganglia will join it in a parallel way so that you get both the gated and the smoothness of the movement to send it to cortex. So you've really got all of your preparations done. Okay? So what are they? Well, basal means down at the bottom, and ganglia means they're collections of neurons. That's all the terminology means. You saw them, I hope, in lab when you looked at the brains that were cut in horizontal and coronal section, and I'll show you both of those today, and then next week we'll go back over those slabs in lab. But we really define them not so much by where they sit. This is a difference than calling something pontine versus medullary. There are fixed boundaries that tell you you're in the medulla versus in the pons. Here it's a conceptual organization about function and connectedness. And so we usually do the defining in terms of what they do in terms of motor function. And as you'll see, the motor piece of it is going to be sort of one multi-step loop. And there will be multiple parallel loops that go not with true motor, but with other sorts of things. Typically, if you have something that you do for motor, what's the other circuitry that you build in parallel for it that's specialized? No? Moving your eyes. Moving your eyes is a special kind of motor. You do separate things almost always for moving your eyes. Remember, you have separate circuitry in the brainstem for moving your eyes, which is really different than any other movement. Okay? And you will have a separate loop for eye movements that comes in basal ganglia. And you will have others about motivational and so-called prefrontal type connections. They are roughly in parallel, but they do intersect so that they're not independent. The major idea that we will have is that we have input from the cortex, and the net output of this whole circuit is back to thalamus. So this is essentially a way of controlling the thalamocortical input from motor systems. So if we're thinking about motor systems, what thalamus do we have to be talking to? V, A, and V, L. And I already told you that most of what are the major input, what are among the major inputs that come into VL? So cerebellar fibers, and so this was cerebellum, and into ventral anterior, I said this, but people probably didn't pay attention, is going to be mostly basal ganglia. Okay? Now, the output is, the goal here is sort of regulating what the cortex does. Not deciding to move one muscle versus another, but how are you going to make that movement? Is it going to be are you facilitating movement or are you defacilitating movement? Are you inhibiting movement? People who are with inhibited movement, anybody have a relative with Parkinson's disease? Okay. See them off their medications, what do they look like? They're stiff. They can't get out of the chair. So the Parkinsonian symptoms are rigidity and slow movements, okay? And that's what's treated with Sinemet or dopamine, L-Dopa, and that's the basal ganglia deficiency disease as opposed to the hyperexcitability disease of Huntington's we'll talk about later. But there are additional structures that interact. We'll mention a few of them. Many of them come up as targets for deep brain stimulation for treatment of movement disorders. And so I want to at least have said the words so that if you encounter them clinically, they don't spook you. So what are the pieces? Well, these are the big lumps, some of which are then subdivided. So we think about a structure called the striatum, okay? It should have stripes. It is striate. And it will be made up of basically three components: the caudate, putamen, and nucleus accumbens. We will mostly talk about nucleus accumbens next week. We will mostly, as our example today, use putamen because most of the motor circuitry flows through putamen. But caudate and accumbens sit in a comparable position in other circuits. In rodents, those of you who've cut mouse brains will know that there is just one structure. They don't have the separation into a caudate and a putamen. They have something called the caudatoputamen. It's not separated by a big bundle of white matter, and the reason is that mice don't have a lot of prefrontal cortex. And that bundle of white matter, as you'll see, is the bundle of white matter going to the prefrontal cortex. So if you don't have prefrontal cortex, you don't need to innervate it from the thalamus, you don't have the big anterior limb of the internal capsule, and your caudate and your putamen are one structure. And we'll see that shortly. Okay? We have the globus pallidus, the pale body, okay? And it has an outside and an inside. This is like easy nomenclature. And then we've got the black stuff, which has a reticulate part, so spiny little cells, and compact cells, big cells. These are the part that are pigmented. This is what makes it black. That pigment is non-enzymatically generated in terms of its initiation, and so it takes time to accumulate over life, so the nigra is not pigmented till late adolescence, early adulthood. Okay. And then we have the subthalamic nucleus. And I mentioned the subthalamic earlier. It's actually part of what we called ventral thalamus. So this is in fact part of diencephalon. So developmentally it is distinct. That's part of the origin of the terminology for it. But together all of these are going to be wired up. And that's what we will then functionally call basal ganglia. So where are some pieces of basal ganglia? Anatomically? Where's the nigra? Midbrain. Okay? So it's not really deep in the center the way we think about it, but we think about it as part of those structurally for this purpose. So where are they? And we'll start with caudate and putamen. Okay? So here is a brain, and what's trying to be shown here is the caudate, and the caudate has a head, a body, and a tail. And this tail loops around. It's curled its tail underneath it. Okay? And this head sits up against the lateral ventricle. So if it sits against the lateral ventricle, and that caudate tail stays against the ventricle, and it curls underneath, where would you expect to find it when it comes around here into the temporal lobe? See, here it is, tucked into the temporal lobe. Well, follow your ventricles back. You follow your lateral ventricle back. You don't go through the foramen of Monroe. You keep going back. You come to the atrium, right? And then you can go from the atrium into the occipital lobe and the occipital horn, or you can move anterior into the temporal lobe, right? And that caudate, which has been along the side of the ventricle, runs now in the roof of the temporal horn, just along the ventricle. It's essentially hugging the ventricle all the way around. Okay? We'll see that it is fenestrated here in its connection to the putamen that sits here as essentially a single giant lump, with these little bridges, and they're actually more than just those few, but through those we're going to have white matter running. And what white matter would that be? We have white matter that runs and fans itself out. Yeah, it's the corona radiata, like this. See that? So those fenestrations that are here, so here's caudate, the head, the body, and the tail coming around. The fenestrations are to allow the white matter bundles to come through. So the anterior portion of that would be what part of what fiber bundle? Yeah, I heard the word. Anterior limb of the internal capsule. Okay? And that anterior limb, as I'll show you in the sections, separates caudate from putamen and a little bit of globus pallidus. So if we think about these and we slice it, okay, here it is. Here's our ventricular system. You can see the lateral ventricle, the atrium, the occipital horn, here called posterior horn, and the temporal horn, here called the inferior horn. Okay? The caudate is immediately adjacent to it. Anterior, adjacent to the lateral ventricle, adjacent to the lateral ventricle, adjacent to the lateral ventricle, but also adjacent to the superior portion of the temporal horn of the ventricle. This is the tail of the caudate, and there will be some point where you can actually catch the little bits of it running back across. So it's a completely long, contiguous structure. You have places where you can see it here as G. You get two views of it, here and here, because it is curved. So in any one coronal plane, you cut it twice. Okay? The putamen doesn't curve like that. The putamen is just this lump that sits, beginning anteriorly, continuing, and then gradually fading out as you move more posterior. And then internal to it, here, we've got the globus pallidus. So if it's pale, what would you expect to be true about it? What makes things pale? So when we think about gray and white matter, why is white matter paler? Because it's white compared to gray matter, which is gray. Myelin. So what do you expect to see more of in globus pallidus than in its adjacent structures? More myelinated fibers. And it in fact has. And as you go from the externa to the interna, it gets paler because you have even more myelinated fibers running into it. Okay? Now, if you think about this, we've got globus pallidus sitting here. We've got some white matter bundles here. What is this? Anterior limb of the internal capsule, and then we've got white matter here that we'll talk about later. And you can think about those white matter bundles like that. And then at the top, you're going to have your putamen. Then you're going to have your globus pallidus externa. And then in here, your globus pallidus interna. Okay? And you can think about this like an ice cream cone. Okay? And the key to understanding the basal ganglia is to remember the ice cream cone and to remember that ice cream comes out by leaking out the bottom. That is the outflow point. And if you remember that, you will understand the basal ganglia circuitry. Okay? By the end of the morning, this will all make sense. Trust me. Okay? But you can see it, and you can visualize it. It's slightly lying on its side, but this is the flavor on the top. Then you've got externa and interna coming in like that. Okay? In this plane, we don't see the substantia nigra. Why not? Where's the midbrain? Well, but I mean, I've got plenty of stuff down here. Where's the midbrain? More posterior. We haven't sliced far enough back to hit the nigra yet. Okay, so where are we? Anterior or posterior? Anterior, what are the clues? Yeah, you got the temporal lobe hanging in the breeze, so it's hanging off in the front, right? Okay, so let's just think about things that we've already named. So this is temporal lobe, makes this, what would that be? Frontal. How about this gyrus right here? And that right there. Yeah, it's gyrus rectus. Here's the hint. That's the olfactory tract right there. That's the gyrus rectus running straight back. You've got that horizontal one. You look at it from the base. You've got to be able to now flip it over in your head. This is why we cut the brains in two planes, and you have one that's intact. You can go back and forth between them and begin to make these into 3 dimensions in your head. Okay? This is lateral ventricles. Okay? This is corpus callosum. This is adjacent to the ventricle, caudate. This is putamen. Down here across the base is where we will find the nucleus accumbens, and I'm going to leave it for discussion until later. What would these fibers be in here? They're part of coronary, but they're also the anteriormost limb of the internal capsule. Okay? Right in here. All right? Here it is on MR. So can you see all of the same structures? Sure can. So here is callosum, caudate. What's that? Anterior limb. This is frontal lobe. Down here is gyrus rectus. You can see all the same things. It's amazing. All right, what level are we at? Yeah, so we move further back. We move more posterior. This is lateral ventricle. This is third. This is row. How about this feathery stuff hanging off here? Choroid plexus. Here we have caudate. Caudate. We have corpus callosum. We have on either side insula. Okay. Here we have putamen. See the boundary right here? That's the boundary. All of a sudden there's a little more white matter, and this is GP externa. Here's the next boundary, and that's GP interna. Those are the contents of the ice cream cone right there. Okay? What's this? And that. Now I heard a mumble. Anybody? Look where it's wrapping. What are these? What's this? White matter tract, it's descending. Yeah, those are the peduncles. So that's actually cutting down just into the boundaries of the midbrain. So what sits and wraps around the brainstem just above the midbrain? We've got optic tracts. Here are your optic tracts coming around. Okay? They have to come around. They come from the chiasm. They come around. They're tucked in there. Okay? Can we see all those same things here? Yes. So here we've got callosum, lateral ventricles, third ventricle, foramen of Monroe, a little hard to see, but that's okay. Caudate, putamen, it's hard to see, but that's probably the boundary to GPE and GPI. Here's the boundary here, GPE. It's a little hard to figure out where the boundary to GPI is there. Okay? This white matter here and here, the optic tracts running back. All right? What level am I at? Give me a nice anatomic landmark that we know from previous sessions. This is the what's this whole region from here? So this is thalamus. So if you're sitting in the thalamus and you've got something, we've just begun to come into thalamus, we're anterior in the thalamus, you've got a reasonably well-defined roundish structure on either side that sits right at the foramen of Monroe on the dorsal medial surface. That is the anterior nucleus of the thalamus. Right below it down here we would have well, it's actually dorsomedial. Comes right to the midline. VA typically doesn't. So this is going to be VA over here. Okay? What is this? Right up against the ventricle, lateral ventricle. Caudate. What's this? Out here. Putamen. What's this? GPE. If you think about the ice cream cone, you've got your flavor at the top here. If you cut and you only see two of them, you're always going to see putamen and externa, but not interna. Okay? All right. So here now we've got, this is internal capsule between caudate and putamen. That makes it anterior limb. This is internal capsule between putamen pallidum and thalamus. That's posterior limb. And so what does that make this region right here? The genu. So what fiber would we expect to find in this region here in the posterior limb? Corticospinal tracts coming down. They come back down in the posterior limb of the capsule. And where do they go? Right into the peduncle. Here we are. And if you cut across right here, what would you see? You'd see peduncle, nigra, red nucleus. Peduncle, nigra, red nucleus, and then right here, you can see a structure that's laid out. It's got white matter on either side of it, just like that. And it's sort of lens-shaped. And that is the subthalamic nucleus. And where is it located? Subthalamic. It's right below the thalamus. Does it look like it should be wired together with the caudate, putamen, and globus pallidus? Not really, but it is. And the white matter bundles, though, come from putamen, globus pallidus externa, and interna, here and back. So they are traversing like this. Okay? Here they are in this plane. It's a little harder to see, but you can see anterior limb of internal capsule and posterior limb. You've got putamen here, caudate here, and thalamus here. It's harder to make out the thalamic substructures on this image. Okay? Which direction have we moved? Anterior or posterior? Posterior, so what's the best new landmark that you see? So you got a lot of thalamus, but there's something new that appeared. What's this? Yeah, it's the posterior commissure. So where does that tell you you are? Are you ventral or dorsal in the midbrain? Yeah, you are dorsal in the midbrain. You've already gone through the ventral midbrain in this coronal plane to come back to the posterior commissure. All right? So we have here caudate. We have over here What's that? Caudate. What's this? Layered structure, looks sort of like Napoleon's hat. Posterior and inferior and lateral attached to the thalamus. Yeah, it's the lateral geniculate. And next to it is the medial geniculate. This is what part of thalamus? Yeah, we're coming into pulvinar. We're probably not quite in pulvinar completely. We've probably in here got VPL and VPM. Here it is. But if we move one more back, where are we now? What's this? What's this? Caudate. And then these little bumps. That's probably caudate caught as it curves around. And on this side, we can actually see a piece of the tail right here. I'm not quite sure what's happening, but right over here, we've got the tail of the caudate. Okay? So we've got caudate, caudate. I just want to make it clear that this is pulvinar, not putamen, with my P's. Okay? What's this? Corpus callosum. Yep. So the posterior pole of the pulvinar is well behind the lateral geniculates. All right. So we've sliced that in that plane. Let's try slicing it in the horizontal plane. Because in order to be able to understand how the connections run, you need to be able to visualize where these fiber bundles sit. So we're cutting now, and we have got anterior at the top of the screen and posterior at the back of the screen. Okay? So let's just get ourselves some basic orientation. So this is corpus callosum. This is, what is it? Remember, the corpus callosum 2 is another one of these curved structures. You cut curved structures in a plane, you can see them twice. So that is the posterior portion. That's the splenium of the corpus callosum back here. Okay? That's connecting this, which would be, what is this going to be? Occipital lobe and parietal lobe with each other. This is connecting frontal lobe with each other. Right? This is caudate. This is putamen. This would be anterior nucleus of the thalamus. Okay? Got to be able to rotate these things 90 degrees in your head. It's tricky. Okay? Caudate, putamen, beginnings of thalamus. Corpus callosum seen twice. We've now slid down. We've got caudate, putamen. This is what part of capsule? Posterior genu. Here is posterior between putamen and thalamus, anterior between putamen and caudate. So again, anterior limb, posterior limb, genu. Caudate, putamen, thalamus. We've now moved inferiorly. We can see putamen, globus pallidus. Okay. Got thalamus. We can't really see much else yet. And now what have we got? What's this? Anterior commissure. It's white matter that crosses the midline inferiorly. What's inferior in terms of our lobes? Temporal lobe. That's the commissure for the temporal lobes. Here we've got putamen, GPE, GPI, and back here we've got thalamus. Here you can see the pulvinar. The geniculates will be back at around this level here. So you can see that there is going to be some pulvinar that will be more dorsal to them. Okay. It's challenging for me even in this plane. Okay? Here we are at the midline. We can see corpus callosum. As we step in, what do we come into? What is this going to be? Caudate. Now what have we got? Arc of the caudate. Here we've got putamen. What's this white matter? Yeah, it's the corona radiata. You can sort of see it just the way that schematic picture was at the very beginning. As you move further back, you're going to have now putamen and then come into thalamus back here with some hypothalamus coming down below. All right, and the last part that we haven't seen yet. So we've seen caudate, putamen, globus pallidus externa, globus pallidus interna, and the subthalamic. And now we have seen the nigra well before, and here it is, down in the midbrain. And the part that we see is the pars compacta. And then the part that we don't see very well is what's known as the pars reticulata. And these will be abbreviated as SNC and SNR. And in general, if you look at them, the pars compacta is the pigmented cells. They are slightly further away from the peduncle, and the pars reticulata is slightly closer to the peduncle. So there's some two substructures here. And that matters because they, in fact, have different neurotransmitters, and they function differently within the circuitry. Okay? So here's the way we do nomenclature. So the whole thing is called basal ganglia. Striatum, as I said, is caudate, accumbens, and putamen. But putamen and globus pallidus, that's the ice cream in the ice cream cone, are known as the lenticular nucleus. Okay? The subthalamic nucleus, to make your lives really miserable in some books, will be called the lentiform nucleus. Not lenticular, but lentiform. Lentiform means lens-shaped. It's also known as the nucleus of Luce. Okay? And the reason that comes into play, this is Luce. Who named it? And I already mentioned one of the trinucleotide repeat diseases when we were talking about cerebellar disease. Dentato, dentate, rubro, red nucleus, pallido, globus pallidum, Lucian, subthalamic atrophy. So that's a disease in which the things that degenerate are things that adjust motor function. Basal ganglia, pallidum, and subthalamic. Cerebellum, dentate outflow, and red nucleus that receives the dentate outflow and also is an intermediate step on the way back to thalamus. Okay? And then finally we've got our nigra, which has both the pars compacta and the pars reticulata. So when we think about the structures, we can now think about where they sit in a circuit. And we'll put that circuit together, and then unfortunately you have to worry about the signs of connections. Because like the example I gave you earlier with vestibular outflow, where you have both a positive and a negative signal onto an important neuron to allow you finer resolution. The whole basal ganglia circuitry is built to allow you to have effective dynamic range with easy adjustment between moving things up and moving things down. So we have our structures. We can think about what the functions are. So the striatum, caudate, accumbens, and putamen, we think about as the input from cortex. Okay? This is the major role it plays. It will be connected in other ways. Basically, information comes in from cortex. Okay? We have a bunch of critical intermediaries in the loop, which are the external segment of the pallidum and the subthalamic nucleus. Okay? We have two outflow points. Remember I said the outflow point of basal ganglia is the bottom of the ice cream cone. That's GPI. That's the major one. The other one that comes into play in some settings is the pars reticulata of the nigra. Okay? They are physically separated, but they have comparable roles. And then this entire circuit is basically tuned by the pars compacta of the nigra. Okay? So We're going to follow things and we're going to march through the various steps, put them together, think about which are inhibitory and which are excitatory, and then have the whole thing make sense. So, the striatum. And all of the pictures I'm going to show you are going to use putamen as a surrogate for any striatal structure. That's because we're mostly talking about motor, because it's where we have the most data, and it's the simplest to draw, because it sits with the ice cream cone. So, we have a cortex to striatum. There are also a thalamic component. This is pretty minor. A substantia nigra component, which is massively major. Okay, we're going to care a lot about that. And then there's a connection from the pedunculopontine nucleus that I think I skipped when we talked about the midbrain, and I think I plan on mostly skipping again today. The only reason to be aware of it is that it seems to be, for people with Parkinson's, for one particular symptom, which is gait freezing. Okay? So people with Parkinson's often will have problems if they encounter a barrier, like a doorway. They get to it and they can't walk through it. They can't initiate the gait. And pedunculopontine problems are very specific for that symptom of Parkinson's. And so deep brain stimulation can be used specifically to treat that, and it targets pedunculopontine. Okay? So we have corticostriate fibers. How are they going to get there? What sits between the premotor, submotor planning areas and the anterior portion of the striatum? You've got anterior limb of the internal capsule. You've got those subcortical white matter fibers. It's basically a straight shot. Okay? We don't see a bundle because they're coming in a very diffuse pattern. They don't make a nice tight bundle and they don't get a name, but they run from cortex to striatum. So where does the striatum talk to? And in this case, we're talking about putamen as our example. And so there's a feedback down to the nigra. That's part of reciprocal connections. But the most important is that it has two outputs, one that goes to GPE and one that goes to GPI. Okay? So short and long. Okay? Now, what did I tell you about the role of GPI? It's an output structure. So anything that goes cortex, putamen, GPI is done. Okay? It's gotten to the output point. Now we've got to worry about what do you do with things that connect into GPE, because we've got to get them to an output. So one of those is that the GPE projects to the subthalamic nucleus. Okay? GPE projects to subthalamic. Subthalamic also gets inputs from the nigra and a little bit from cortex. But this is the big one that matters. Okay? So now we've gone cortex to putamen, putamen to GPE, GPE to subthalamic. What do you think is going to come next? Let's think about outputs of the subthalamic. So, goes mostly to GPI. We've now gotten our information to the output device. So we went cortex to putamen, putamen to GPI. That was direct. And then we went putamen to externa to subthalamic to interna. That's an indirect way to get to the output device. It also projects to the pars reticulata of the substantia nigra. What's that? Another output device. Okay? So, if we think about those globus pallidus connections, we've got GPE to the subthalamic to the GPI, and we've got innervation back and forth from the substantia nigra. Okay? That's all set. But we haven't gotten anything out of GPI yet, right? We're now stuck with information at GPI, and we want to get it out. And where did I say all of this information goes? To the thalamus. So GPI goes to thalamus, and it's in fact going to go specifically more to ventral anterior and a less to ventral lateral, whereas cerebellum goes more to ventral lateral and less to ventral anterior. And it's going to send it in two separate bundles. There are some that loop around the subthalamic fasciculus, that's this dotted bundle here, and others that just loop and go on the dorsal surface. So some are going ventral to it, and some come dorsal, and then they loop around this structure here and are met by fibers that have come from the contralateral dentate. So remember, subthalamic sits just above, if I go quickly back, like 45 slides. I need to move this picture in there later for next time. But if I come back to where I saw the subthalamic, okay? So here is GPI. Here's the subthalamic. Here's the red nucleus right here. That red nucleus got fibers from where? Cerebellum, contralateral dentate. Someplace over here, came out, crossed, wrapped around. They're continuing on up to the thalamus, just like this, right? So we've got these GPI fibers that are coming out. Some are coming around the subthalamic, some are coming dorsal to the subthalamic, and they're all turning here, and they're all joining with the cerebellar fibers to come into the motor thalamus. Okay? So that's where they are. Let's jump way ahead. Oops. All right. So here we've got the thalamic fibers that are coming as well. Where's my point? Where's my pen? There we are. So here we've got the cerebellar fibers that come as well, and these all come into thalamus together. We've got the fibers that go ventral to the subthalamic, which is known as the ansa lenticularis. It's like the ansa cervicalis. It makes the loop. Okay? So we've got fibers on either side, and then we've got the fibers that go back and forth between pallidum and subthalamic, which are known as the subthalamic fasciculus. Okay? So we have the ansa lenticularis, the lenticular fasciculus, which are these fibers, and then when it joins with all the others, we call it the thalamic fasciculus. So the thalamic fasciculus has cerebellar and basal ganglia. The lenticular and the ansa have basal ganglia going to thalamus, and the subthalamic fasciculus has fibers that go back and forth between globus pallidus and the subthalamic nucleus. Okay? Terminology is really important if you're going to try and describe what fiber bundle you're trying to put an electrode into to treat somebody with Parkinson's disease. Okay? Now, how about the nigra? Well, the biggie is the nigrostriatal projection. This is the dopaminergic projection that comes from the substantia nigra into striatum, and striatum is the shorthand for caudate, putamen, and accumbens. Okay? It gets reciprocal connections from both putamen, from striatum, and from globus pallidus. And then it is reciprocally connected, particularly the pars reticulata, with the subthalamic nucleus. Okay? We'll skip the pedunculopontine. So, when we think about these loops, you need to now think about ways synapses work. So, some of them are excitatory and others are inhibitory. Cortex output is excitatory. It's excitatory whether it's a descending tract to the spinal cord, whether it's going to thalamus or going to striatum. Okay? The subthalamic is excitatory, and then the cortex back to the thalamus back to the cortex is excitatory. But most of the others are inhibitory. So the striatal outputs, the putamenal projection neurons, are GABAergic. The GPI and GPE neurons are GABAergic, as are the pars reticulata, which is just like the GPI. Okay? So now let's think about how we can divide this in two. Okay? So what I'm going to teach you here is what's known as the classical model of basal ganglia. It's actually now 30 years old since it was published. The nicest early review paper to look at comes from, actually from Ann Young, it's Ann here, and Jack Penny next to her, Roger Albin was their fellow at Michigan when they were the movement disorder people there. And they basically figured out how the basal ganglia worked by knowing anatomy and then figuring out what the neurotransmitters were and what transmitter receptors were. And then when they drew out the schematic, they finally understood how not only the circuitry works, but how Parkinson's disease works and how Huntington's disease works. And it still is essentially the fundamental way we understand basal ganglia. Everything beyond that is sort of window dressing on top of it. Anne went on, was chair of neurology at MGH for about 20 years, stepped down about 8 or 9 years ago. Jack, who was also her husband, who's there, the two of them went to Lake Maracaibo in Venezuela many, many times to study a massive kindred with Huntington's disease, from whom the gene was first mapped and then eventually cloned. That's Jack down there examining a small child with early stage Huntington's, asking them to stick their tongue out. He died very, very young, unfortunately, about 20 years ago. But anybody who wants to sort of understand the sort of thinking behind this should go back and read their paper. It's really a beautiful layout of how you think about things based on how you put anatomy and function together. So we think about a direct pathway. It goes cortex to putamen to GPI and out. That's the shortest loop you can build here. You can't go from cortex to GPI without stopping in the putamen. Okay? So we think about that. How does that work? Well, when you do it, your cortex drives a neuron in the putamen. You drive that neuron, but it is an inhibitory neuron. So it inhibits your GPI neuron. If you inhibit your GPI neuron, what happens in your thalamus? Does activity go up or down? You got a 50-50 chance here. It goes up because you're inhibiting one of its inhibitors. So this is a pathway that increases corticothalamic feedback. Okay? Let's look at the indirect pathway. We start with the same drive from cortex to putamen. So it drives this neuron here. This neuron is inhibitory. So it inhibits its target, which is a neuron in the externa, which is inhibitory. So we decrease the firing of this neuron. This neuron is what? It's an inhibitory neuron. So we are reducing the inhibition in the subthalamic nucleus. So what happens to the subthalamic nucleus? It becomes more active. It is a glutamatergic neuron, so it drives the neurons in GPI. And what happens to the thalamus? We increase inhibition. So by adding the extra synapses and dependent on the signs of the neurotransmitters, you come up with a pathway again that allows you to very much fine-tune. You have a way to turn it up and a way to turn it down. You're not dependent on systems where you can just turn it up and wait for it to naturally quiet down on its own. Or you can quiet it and wait for it to come back on its own. You've got the ability to turn this knob in both directions. And that's what you need in order to generate facile motor movement. You need to be able to make that just right. You need to be able to tune your motor activity for the situation you are in. Okay? So, direct and indirect. It's a big idea. You got to really get it. Thalamus drives the cortex, but GPI inhibits the thalamus. The direct striatum inhibits GPI, increases thalamic activity, is a positive system. Indirect, striatum inhibits GPI, which reduces the inhibition of the subthalamic, which drives the GPI, which quiets the thalamus. Yeah? Means that thalamocortical fibers are glutamatergic. They're excitatory. It does, but if you don't have thalamic activity, you don't hear anything. So that the basic goal is to provide thalamus drives your cortex. It tells it something is going on. If the thalamus is not firing because the neurons are inhibited, you get no useful information in and you don't respond. You are not aware of something. Okay? So Direct increases activity, always. Indirect decreases activity, okay? But now the question is, how do you balance these, okay? And the issue is, this is the role of dopamine from the nigra. Okay? So the nigrostriatal pathway is going to tune between direct and indirect. Okay? So you've now got a very fine balance based on how much those neurons fire as to which way you are tilted. Are you more towards the indirect and low activity, or more towards the direct and more activity? So the dopaminergic projection, we'll talk about dopamine in a bigger context at the very end. But it is a neuromodulator, not a synaptic, not a transmitter in the way we typically think of it, in that it basically uses not the ion-gated channels in the similar way, and it has longer lasting, and it will actually give you a much more broad effect. We lose this in Parkinson's, and the real key question is, how do you know the difference between direct and indirect pathways? Okay? So if we draw the map like this, we've got our motor cortex talking to our neurons sitting here in the putamen, some of which go directly. So this is a neuron in the putamen that goes directly down to GPI. Which pathway is this? Okay. And here's one that goes to GPE to subthalamic, and this is therefore indirect. All right? So we're driving both of them. How do you adjust this? And the key is that the putaminal projection neurons that project either to GPI or GPE express different dopamine receptors. So when you release dopamine, you normally are driving them in opposite directions. So dopamine drives the direct, okay, and inhibits the indirect, okay? So what does dopamine do? You increase direct and decrease indirect. What are you doing? Are you facilitating movement or inhibiting movement? You're facilitating movement. So the balance is through this distinct type of dopaminergic receptor. And there are multiple types of dopaminergic receptors, and there's a little complexity here, but in general, they're either inhibitory or excitatory, and this is what gives you the balance. If you lose the dopamine, what happens? If I take away dopamine, what happens to the direct pathway? It goes down because dopamine is driving it, okay? And what happens to the indirect pathway? It goes up because you remove the inhibition from dopamine. And now you have a balance where you've got more in indirect pathway than direct pathway. And what do you end up with? Decreased movement, slowed movement, difficulty to initiate movement. That's Parkinson's disease. And Parkinson's disease is marked by the degeneration, among other things, of the nigrostriatal dopaminergic neurons. And so with understanding the circuitry, you actually have complete physiologic understanding, at least at the first level, of Parkinson's disease. So Parkinsonism is the syndrome of bradykinesia, rigidity, and tremor. And we're not going to talk about where tremor comes from because it's the most complex part of anything that I know. And really no one knows where tremor comes from. It's really interesting. It's a very interesting topic, not for us. If you give somebody an anti-dopaminergic drug, you will block this pathway. And you can make somebody Parkinsonian, not because the cells are degenerated, but pharmacologically. Okay? You can have it for a variety of other diseases. It can be a component of other diseases, but most of what we see clinically is what we call idiopathic Parkinson's. It's Parkinson's that arises without a point mutation or a genetic cause or something like that. The histologic component characteristic of typical idiopathic Parkinson's disease is this inclusion here. This is a nice, smooth, pinkish inclusion. You can get a little sense of a halo. What's all this black stuff? Well, we're in the substantia nigra. I mean, it's got to be nigra for something, right? That's pigment in the neurons. And this is essentially a melanin-like pigment. It actually has many of the same characteristics as cutaneous melanin, except it is not generated the pathway to make it does not begin with tyrosinase, but rather begins with oxidation of dopamine that is present in these cells. Why is it present in these cells? They use it as their transmitter. So they are making lots of it. And then once you've got the oxidated product, the subsequent processes can turn it into eumelanin and I can't remember the name of the other cutaneous pigment. Anyway. All right, so this is a Lewy body. This is Lewy, right here. This is Lewy when he was a student. This is Alzheimer, who was his professor. And we now know that this is, in fact, made up of a protein called alpha-synuclein. And, in fact, point mutations in alpha-synuclein cause familial Parkinson's disease. It's an aggregating protein. It propagates. You may have read papers about, can you get this from eating contaminated food? There's a whole complex argument about whether this needs to be thought of like a prion disease or not. It's interesting, Lewy didn't describe them in the nigra. He described them actually in the basal forebrain. All right, let's take a break because I could use a little bit to rest my throat. And we'll pick up again here, and we'll go through some of the white matter bundles and talk a little bit more about the function when we pick up. Let's pick up again in about 10 minutes at 20 of. All right, let's get going again. So I scribbled this on the side of one of the pictures before, but this is the terminology for white matter bundles that we have to be aware of, and the names get very similar in lots of ways. So first of all, we have our subthalamic fasciculus. This is essentially the wiring of subthalamus back and forth to globus pallidus. And in fact, although we can draw it as a back and forth, it's actually really from GPE to subthalamus to GPI. There are tiny little connections that go in the other directions, but this direction of information flow is part of the indirect pathway, pallidum to subthalamic to pallidum. That's the critical piece. We have two fiber bundles that are leaving globus pallidus interna from the bottom of the ice cream cone on their way to the thalamus, some which go ventral to the subthalamic and some which go dorsal. And when you think about the subthalamic nucleus, when you see it in the coronal plane, it's lens-shaped like this, and it's got white matter on this side. That's ANSA on the ventral surface. And on this side, it has the lenticular fasciculus, and those are the white matter bundles that wrap the subthalamic. Okay? They will then have to turn around, and when they do that, they are joined eventually by the fibers that came from the contralateral dentate, synapsed on this side, some of them at the red nucleus right here, but others continued on and are other fibers going to motor thalamus. And at that point, when we've got ANSA and lenticular and cerebello or dentatothalamics, we can call the whole thing the thalamic fasciculus. But notice that the subthalamic fasciculus does not feed into the thalamic fasciculus. Okay? So, if we look at those, there is this dotted line here, which is harder to see. That's the ansa lenticularis. There is the lenticular fasciculus here, labeled up here. That's on the dorsal surface. Those are on either side. Between them sits the subthalamic nucleus. They're very separate from the subthalamic fasciculus, which is reciprocal between subthalamus and pallidum, and pallidum. Okay? Now these make this loop and will be joined by fibers that eventually have come, and at this point we will have gained fibers that came from cerebellum. There are a series of so-called fields of Forel. There's the field H. H is where you've got the cerebellar fibers coming in. And then there are the two fibers bundles that are on either side of this little bend-around zone where we have a gray matter structure known as the zona incerta. And these are called H1, more dorsally, and H2. H2 is equivalent to what fiber bundle? The lenticular fasciculus. Okay? Labeled all the way up over here to make things confusing. Okay? So we've got field H1 and H2 and then H. Okay? Those are the fields of Forel. It's basically by the time you come into it, you're beginning to become thalamic fasciculus. This is Forel. He's a Swiss neuroanatomist. Worked at the Institute of Psychiatry in Zurich. You know, it says something both about him, perhaps, but more about the Swiss, that they made a stamp with him on it. The probability of the U.S. having a stamp for a neuroanatomist on it is probably pretty low. Okay? But in addition to having a stamp, this is, in fact, this is from Google Maps. This is the Institute of Psychiatry in Zurich, and it's on the street that's named for him. Okay? August Forel-Strasse. And what does that make these? Yeah, those are the fields of Forel. Okay. You ever going to forget the fields of Forel now? No. You want to bet? I have an easy way to find out. Okay. All right. So what's the stuff that's in between? There's this area that it bends around. It's called the zona incerta. It's the stuff in between. It's a really poorly understood anatomic area. But what's interesting is that it's wired incredibly tightly to an enormous number of things that we already know about. So it's wired to motor and sensory cortices. Okay. It's wired to thalamus, to colliculus, pedunculopontine. Remember, that's that structure I talked about for gait, basal ganglia. It's wired into the superior colliculus in multiple places. Brainstem sensory nuclei. It is some sort of important central integrator. It sits really right near lots of things that are passing by, and it basically picks up inputs from them. We actually begin to know a little bit about it these days because as people treat Parkinson's and other movement disorders with deep brain stimulation, as you are placing the electrodes, if you go through the zona incerta, why might you go through the zona incerta? Well, say you're putting your electrode in and you're trying to get to the subthalamic nucleus. On your way in, you might start to record and go, oh, I now anatomically know I am here and I can recognize what the pattern of activity is in H1. I'm here and I'm in zona incerta. I can know what the pattern of activity is in that. I'm here in H2. Now I have just passed into the subthalamic nucleus and that's my target. My electrode is in the right place. So in fact, knowing the physiology of the different components is really important for guiding deep brain stimulation electrode placement. So this is not just sort of abstract, but it's actually in use every day clinically as people are being treated for diseases. So we built this loop for the motor system. We brought in our motor input from the supplemental motor area. We will see later that we bring other areas in. But it went to putamen, it went to GPI and pars reticulata, and it actually goes to the ventrolateral portion of GPI. Don't worry about this substructure. And then it goes to the thalamus, okay? And then the thalamus comes back to cortex. But there's a separate loop that does oculomotor. So what eye field would you use? Well, it would be the frontal eye fields. That's sort of the planning eye field movement motor component comparable to the supplemental motor. Tends to use the caudate. It tends to use a more centrodorsomedial portion of GPI and a little bit of reticulata, and it goes to a slightly different part of thalamus. So we've got two loops right there. And remember when we talked about prefrontal, it was a lot about behavior and motivation. which in many ways is really like motor activity. It's about your output level. Are you apathetic or engaged? Are you making decisions or just doing whatever happens first? That's a sort of hypermotion type response rather than think, then pausing and thinking. So that there are loops that come from those dorsolateral prefrontal areas. The dorsolateral prefrontal cortex here just called DLC, which is usually DLPFC, uses a different part of the caudate, a different part of globus pallidus, different part of thalamus, and loops around. But the same structure. We know that this is involved and important for executive function and problem solving. So basal ganglia lesions can lead to disruptions in those because they change the gating of it, not because they damage the sort of critical structures. We know that the orbitofrontal regions were much more about personality and civility, understanding context, socially appropriate behavior. Think back to the example of Phineas Gage. You know, he curses inappropriately, which he never did before. And that goes through a different part. But again, the same circuit of cortex, striatal, palatal intermediates with the subthalamic, palatal internal and reticulata outputs, and components of thalamus and back. And then finally, we'll see that we have a loop that comes from the anterior cingulate to the so-called ventral striatum that we'll talk about when we talk about hypothalamus and cingulate structures, that uses other components of GPI as well as other components in thalamus, in fact. And we know that anterior cingulate is connected with both the dorsomedial nucleus as well as the anterior nucleus of the thalamus. And here, you're gating not so much physical motor, but motivation. And so the difficulty that you get with it is apathy. You get that loss of engagement, that sort of bradykinesia of emotion, in a way, to sense it. Okay? So one of the common components of Parkinson's disease is depression. Okay? And that probably is, in part, these circuits being manifest as depressive symptoms because you are essentially slowing them down as well. And you can use deep brain stimulation to try to treat depression in a manner similar to what you use to treat Parkinson's. You will have different targets, but it's basically the same thing, which is the concept is you want to disrupt this circuit which is currently inhibiting your activity too much. Okay. So it is simultaneously doing all of them. There are separate things that it does in anterior cingulate where it is much more driven about context and reward. And nucleus accumbens dopamine is different. It comes from not so much from Niger, but from VTA, and that's got much more context-specific control. Yeah, but we'll talk about this, and you'll hear more about it at the very end of the course in the lecture on sort of reward and motivation. All right. There's a little bit of additional complexity in here that came about when people were staining. So when you're trying to understand structures, you look at sections and you stain them for various things. And you stain them for anything that you happen to have a reagent for, okay? And so this was staining for acetylcholinesterase. Now, why would you stain for acetylcholinesterase apart from the fact that you have an assay? Well, we've talked about glutamatergic and GABAergic neurons and dopaminergic neurons. Well, there also turns out to be cholinergic innervation of these structures. And there's cholinergic, at the very end of the course, we'll talk about these diffuse projecting systems. But when this was done, and this was work actually done by Anne Grabiel when she was actually a postdoc in Nauda's lab. She's now an institute professor in brain and cognitive science at MIT, having worked on basal ganglia, having started as a grad student for Nauda early on. What she saw were there were areas that had lots of staining and areas that had less staining, and that these were sort of irregular patches. They were named matrix and striosome, stria being the abbreviation for striatum. And there's some difference in what comes in and what goes out of them. We still don't have a really good handle on how they function differently, but they must, because we know that what we typically think about as the sort of globus, as the basal ganglia circuitry, is what happens in the matrix. What goes on in the striosome is something else. Some of it is that limbic component, but some of it probably is other circuitry that we don't understand. And the problem is that you can only identify where you are if you stain in this manner. So that in an intact animal, when you've passed your electrode, it's very hard to know where you are. Okay? And as you move deeper and deeper and deeper, you'll go in from one into the next, out and back and forth. So it becomes an enormously complex problem in trying to reconstruct anatomically where your physiology came from. And I'll leave it at this point. John Assad will talk a bit more about this when we discuss motor systems in the next block in review. There are distinct neurons here, and it is in the GPE, the medium spiny neurons are the projection neurons. They're spiny because they have lots of dendritic spines, okay? And these, in fact, are the first cells that degenerate in Huntington's disease, as I'll show you in a moment. So if you have lots of spines, what is that? What are spines used for? Yeah, they're inputs. They're regulatory input devices. By changing the shape of the spine, you can change its ability to have current pass from the spine down into the dendrite. You talked about this in the physiology block, right? Positioning of them influences how strong they are. And most of the, as you'll see here, the acetylcholine and the GABAergic inputs are almost always cell body. The glutamatergics and the dopaminergics, which regulate, are out on those dendritic spines and on the big dendrites, okay? So that every time you have a cortical input on a spine head, it is being regulated by that dopamine. And what does that dopamine do? Well, it differentially regulates direct and indirect pathways. Synapses in exactly the same location on neurons in the two different pathways. But based on where they're projecting, it also determines which isoform of dopamine receptor they express. So here's an incredibly interesting developmental biology problem. How do they know how to do those coupled? Okay? You have different axon growth pattern, and you're going to express different dopaminergic receptor subtypes. They better be linked somehow, okay? Because you don't want to have a mixture of the wrong dopaminergic ones going to the wrong pathway, because then you lose the ability to regulate. So there's interesting, you know, basic neuroanatomy prompts interesting underlying neurobiology. So, which is the normal and which is the abnormal? These are two different people. Where are we? Anterior, posterior? Anterior. How do you know? Yeah, shape of the temporal lobe. Okay? So then we've got, down here, this is the temporal lobe, right? This is corpus callosum. This is corpus callosum. What do you think about it? Does it look the same size as its friend on the other side? Bigger or smaller? Yeah, it looks small to me. All right? This is, in here will be nucleus accumbens. This is anterior limb. All right? What do you notice here? Well, here's the anterior limb. So caudate is small. Putamen is small. Both of them are very small. In fact, this is not as end stage as it gets. This is Huntington's disease. Okay? So Huntington's disease is a movement disorder marked by chorea. What's chorea? So involuntary large movements, not just twitches, but big movements, uncontrolled big movements. Okay? So that's sort of the opposite, in a way, of Parkinson's, where you don't make any movements. Here you're involuntarily making big movements. Pathologically, it's a trinucleotide repeat. It's an expansion of a poly-CAG domain, which is translated and gives you a long polyglutamate repeat. Those aggregate, and we can actually stain for them, and you can see them as these large aggregates in neuronal nuclei here, here, here. They were, in fact, missed by neuropathologists for a century until we had a good stain, at which point you could go back and see them. And so until the biology was done in Jim Gazella's lab in the Navy Yard, we didn't have a good way of sort of doing the histologic diagnosis. We now know this. The nuclear inclusions probably play some role in pathogenesis. They actually bind a series of transcription factors. They probably lead to some degree of transcriptional dysregulation. But it's still very unclear what it is that drives cell specificity in terms of degeneration. But the cells that degenerate, we think about the populations that are in the striatum, in the putamen and in the caudate. We've got our projection neurons and our interneurons. And the ones that degenerate first are the encephalinergic, GABAergic projections to GPE. So what does that mean? What pathway is GPE part of? Indirect. If you lose them, what happens to information that you're trying to shove through your indirect pathway? It doesn't get anywhere. So what happens? You have much more direct. What do you end up with? More motor activity. Okay? The second ones that degenerate are the ones that project from putamen to GPI. So later in the disease, what's going to happen? They're going to typically become rigid. Okay? And so if we start in this normal circuit, and we've got we've separated them out not based on D1 and D2 subtype, but on direct and indirect. The first ones we lose are these, and they're also marked. They use different co-transmitting peptides. They use enkephalin rather than substance P. You decrease that GABAergic signal to the GPE, you decrease and you allow it to inhibit, you make everything go too fast. Okay? Over time, you lose both of them, and then you decrease your output back to the cortex, and you end up with a low motor output state. Okay? What do you see? So there's lots of anatomy we could do here. So let's start off with, what's this? Corpus callosum. This is, that's caudate. Yeah, caudate on the other side. This is putamen. This would be posterior limb, right? Okay. This is here and here. Well, this is the white matter. Right here is something that's like got a little black line running through it. Yeah, that's our nigra. This is red nucleus. This lens-shaped thing is the subthalamic nucleus, right? The white matter on this side would include the ansa, and on this side, lenticular, and then as they turn around, they become the thalamic and come right here to the thalamus. Okay? Here it is on this side. What's wrong? Yeah, there's some bloody. Where is it? Is it in? Yeah, well, more than pallidus. Be specific. It's exactly in the GPI. In fact, it has taken out exactly the entire GPI. Does that look like something that happened naturally? No. This is, in fact, a pallidotomy. This was for about a decade and a half one of the best available treatments beyond dopamine replacement for Parkinson's disease. And the goal was to take out the GPI. What happens if you take out the GPI? What is the GPI doing? Which pathway is it part of? GPI. Both. So you take it out. What do? You remove inhibition on the thalamus. Okay? And you increase the ability to move. And these were reasonably successful. This person actually died 48 hours after his pallidotomy from a large PE from unrelated causes. but lots of people got significant amounts of benefit from it for a reasonably long time. But the problem with it is that it's a, if you think about the pathways, you've got your, put the microphone back before I lose my recording. You've got your direct and your indirect, and if you take out the subthalamic, or you take out your GPI here, you're going to basically allow the thalamus to be more active and have greater thalamocortical drive and to let your motor activity come through. But it's not adjustable, okay? So what's done now instead is to place stimulating electrodes in towards the subthalamic nucleus and stimulate it. Now, in fact, what you're doing is not trying to drive it physiologically, because what would happen if you physiologically overdrove the subthalamic nucleus? What does the subthalamic nucleus do? Projects to the GPI. What type of synapse does it make? Excitatory. So if you drove that, what would you do? You would drive GPI, which would do what to your thalamus? Inhibit. Is that what you want? No. So in fact, what you're really doing is essentially causing depolarization block by stimulating it at a frequency and strength such that it can't fire. So you're essentially taking it out of the circuit. Why is it better than just taking out the GPI completely with a lesion? Because you can fiddle with it. You can tune it so that over time, as pathways change and have plasticity and other things take place, your nigrostriatal pathway is essentially continuing to degenerate. You can adjust what you need from it. And so the problems are that the parameter space is massive. How much frequency, sort of how much current you pass and the frequency you deliver it at is a huge space that you could try and search. And it takes a couple of hours for somebody to physiologically settle into the new state. So it's very hard to know how to search it functionally. It's done, there's small little tweaks. And actually at the General, Alice Flaherty is the neurologist who mostly manages these people. Alice is a HST MD PhD grad. She actually did her PhD in Anne Graybiel's lab, understanding basal ganglia physiology. But it's a really complex issue. So a lot of the lesioning issues had to do with what's nearby. So that when you go, if you want to make a lesion and you want to have it contained, to a large extent, you're basically using current and thermal heating to kill it. And the way those lesions were made, white matter helped contain that. If you come down and you think about it, the subthalamic is much closer to the posterior limb of the capsule. And if you had your lesion spread too far, you could make people, you give people motor weakness, as well as disrupt red nucleus on the other side of it, and then all of the cerebellar fibers. So that the bulk of the rationale, they're equal targets, but for creating lesions, it was thought that pallidum was safer. People had been doing thalamic lesions, not particularly successfully, for pain management and the like, and tried it for Parkinson's as well. But the choice of targets, and there's a whole massive neuroscience literature now about, is subthalamic the best place? And in fact, if you pass your electrode and you get to it, and you have an electrode that's here, but it actually traverses other structures, if you put other recording and stimulating points on it, you can consider, do I want to stimulate with a circuit where my current flows like this, or where my current flows like this, or like this? And you can acquire physiology data from all of them as well along the way. So these are done typically with awake patients. So they have, the burr hole is made, the skin is anesthetized, the bone is anesthetized, the burr hole is made, they are awake, and they're being asked to do motor tasks. So they're being asked to try and do motor tasks while the recordings are being done. There are electrophysiologists looking at and listening to them. Then when they get the electrodes in, if they turn them on, and you'll see videos in the next block, when you turn somebody's stimulator on, they can be sitting in the chair, unable to get out of the chair, tremulous like this, just unable to stand up. They turn the stimulator on so the stimulator has a battery pack that's usually in the chest. It's got an electromagnetic control to turn it on. You turn it on, the tremor stops, and they can stand up and they can walk. Okay? I mean, it's magic when you see it happen. Okay? So they do this awake with patients in the OR. They watch their tremor, they watch their movements, in part to help them know where they are. But the other is that, and you may hear about this in the next block as well, the major other issue is, right now the stimulators are on all the time. They're at a fixed frequency, at a fixed current, and they're always on. But you're not always moving. Okay? Wouldn't it be nice if the stimulator only came on when you wanted to make a movement, and it allowed the movement to happen, so that when you're lying in bed asleep, you're not stimulating? Why does that matter? Well, the biggest issue for these people is they need their batteries changed. Okay? These things don't run on magic. They run on a battery. And if the battery is running all the time, 24/7, but you're not moving 24/7, and you only need the circuitry running when you're moving, then you could turn it off when you could have low level or high level. You could modulate it, and you could lengthen battery life. So right now, these people get their batteries changed roughly every 9 to 12 months. But it also means that you need to stay someplace where you have access to somebody who can change your battery and adjust your parameters. And so systems that are that are closed loop, that monitor cortical activity and cerebellar function, and say, getting ready to make a movement, gate the basal ganglia, which is what you physiologically do, okay, would be ideal. And that's a major issue of how you gather those signals, process them, and feed them back, all in a device that you could implant. All right, I'm going to stop there. I'm not going to talk about the vasculature over the course of the weekend, assuming that I have electricity at home again. I will make a full vasculature, which will cover the vasculature from Wednesday, which is the anterior circulation, so circle of Willis, middle, anterior, and posterior cerebral arteries for cortex, as well as deep circulation, first for thalamus, and then for basal ganglia. And at that point, we'll have sort of covered all of the vasculature. And so I will try and get that put together. I'll try and get it made tomorrow and up at some point over the weekend. And you should just look at that on your own at some point. We talk about vasculature in lab on Monday again when we look at coronal slabs. So at that point, we'll pick up there. And I'll see you Monday, hopefully feeling better.